Good morning, dear colleagues. My name is Anna Malchikova. I am mycologist in a laboratory of clinical microbiology, mycology, and antibiotic treatment of National Research Center for Hematology in Moscow, Russian Federation. And I want to thank the organizers of the International Conference on Microbiology and Infection Disease for the opportunity to present you the results of our study in vitro activity of antifungal drugs against biofilms and planktonic forms of Candida species isolated from blood cultures in patients with hematological malignancies. The authors have no conflicts of interest to declare. Candidemia is a severe clinical complication in patients with hematological malignancies. In 2016, the European Organization for Research and Treatment of Cancer published a study in which the frequency of candidemia in this group of patients varied from 1.5 to 0.4%. In a population-based study conducted from 1990 to 2019 years, the incidence of candidemia reduced to 3.3 for 100,000 people, but a 30-day mortality rate increased to 0.4 per 100,000 people. At the same time, a higher mortality rate reaching from 40 to 50%, longer hospital stay, and higher costs allow us to consider candidemia as a severe complication in patients with hematological malignancies, which requires special attention. Recently, the rate of biofilm-forming Candida species has been increased. Maria Tumbarella demonstrated the increasing of the rate of Candida biofilms from 20 to 2004 years, while the population of proportion of Candida non-albicans biofilms increased 2.5 times from 90 to 47% over the same period. In Russia, the increase in the frequency of detection of Candida biofilms was demonstrated in a multi-center study on the basis of the National Research Center for Hematology. 428 patients with invasive candidiasis were included in this study. Biofilm forming ability was predominant among Candida non-albicans and increased from 45% to 60% in 2005 to 2007 years, to 60% in 2016 to 2017 years. We found a surge in biofilm production, which can be explained by the use of invasive devices for which Candida had more tropism, or by a more severe patient condition during this period.
Sorry, doctor, we cannot see your slides. I don't know if the others can see, but for me, I can see only the first slide only.
I'm sorry.
Yes, now, yes.
And now?
Yes, now I can see the slide. Thank you.
And now?
Yes, now I can see slide 5.
5.
Thank you.
Oh, okay. The ability of Candida to form biofilms on invading medical devices complicates the management of candidemia. Italian and Russian scientists demonstrate the significantly higher mortality rate in patients with biofilm-positive candidemia compared to biofilm-negative episodes. There is no statistically significant among in other reports. Moreover, recent evidence suggests that zolbas therapy is linked to an extremely high mortality rate of about 70% in patients with candidemia caused by biofilm-forming candida. In the human body, candida biofilms can form on all invasive devices, but in patients with hematological malignancies, central venous catheter is the most frequent device in which candida can form biofilms. The ability of candida to form biofilms depends on the material of the invasive vascular catheter. In experimental research, it was proven that candida rarely formed a biofilm on polyurethane and silicone, but it was often formed on latex, Teflon, and polyvinyl chloride. According to different studies, biofilm production differs among causative agents of candidemia and varies from 20% to 17%. In Russia, this figure is 42%. Biofilm production among different species of Candida is not the same. According to our research, the main biofilm production among Candida species causing invasive candidiasis were Candida tropicalis, 90%, Candida cruzi, 75%, follow as Candida parapsilosis, 41%, albicans, 29%, and glabrata, 27%. Life cycle of Candida biofilms includes four stages: inherence, initiation, maturation, and dispersion. It is extremely difficult to influence the biofilm on maturation and dispersion stages. And not all groups of antifungals are active against biofilms in these studies. Candida biofilms display reduced in vitro susceptibility to the most antifungals generally used for candidemia treatment. There are several studies evaluating in vitro antifungal susceptibility to Candida biofilms. It was demonstrated that fluconazole and voriconazole and conventional amphotericin B were not active in vitro against Candida biofilms compared to planktonic forms. In contrast, echinocandins and lipid formulation of amphotericin B showed activity against Candida biofilms. It is quite certain that additional studies of their antifungal susceptibility are extremely important for the adequate management of patients with candidemia. The aim of our study was to evaluate the in vitro susceptibility of anidolafungin, caspofungin, fluconazole, and amphotericin B conventional form against biofilms and planktonic forms of Candida species isolated from blood culture in patients with hematological malignancies. Biofilm formation among Candida species was assessed with XTT method proposed by Pierce et al. Antifungal susceptibility on planktonic forms of Candida species was determined by broth microdilution method as described by Clinical and Laboratory Standard Institute M27 methodology. The minimum inhibitory concentrations of antifungal drugs for Candida species biofilms assessed using the XTT reduction assay. During 2006 to 2018 period, a total of 75 Candida isolates were evaluated, from which were 26 Candida albicans, 15 Candida parapsilosis, 9 Candida cruzi and Candida tropicalis, 8 Candida gallermondii, 3 Candida lusitania, 2 Candida glabrata, 2 pelliculosa, and 1 Candida kefir. Biofilm production was detected in 45.3% of Candida isolated, from which were 89% of Candida krusei, 78% of Candida tropicalis, 33% of Candida paracelloses, 27% of Candida albicans, 35% of Candida collermondii, and no one of Candida glabrata isolates were produced biofilms. Antifungal susceptibility was available for the main Candida species, account 27 isolates, caused candidemia and formed biofilms. According to the CLSI interpretive breakpoints, all planktonic forms of Candida were susceptible to anidulafungin. Caspofungin demonstrated lower activity against planktonic forms of Candida species compared to anidulafungin. The rate of susceptible isolate was 85%. 4 isolate of Candida krusei were non-susceptible to caspofungin. 1 isolate was resistant to caspofungin with minimal inhibitory concentration for 1 microgram per milliliter, and 3 isolate intermediately susceptible to caspofungin with minimal inhibitory concentration 0.5 microgram per milliliter. All planktonic forms of Candida species are characterized as being wild-type to conventional amphotericin B. The susceptibility of planktonic forms for Candida of fluconazole was relatively lower than for other tested antifungal drugs, 67%. 1 isolate of Candida tropicalis was resistant to fluconazole with minimal inhibitory concentration, 8 microgram per milliliter. Minimal inhibitory concentration of anidulafungin and caspofungin were similar for both forms of Candida, such as biofilms and planktonic forms. Minimal inhibitory concentration of fluconazole were 1,000 times higher for Candida biofilms compared to planktonic forms. For conventional forms of amphotericin B, this parameter was 16 times higher. Fluconazole exhibit no activity against all species of Candida biofilms. Conventional amphotericin B was found to have lower inhibitory effect against biofilms of each tested Candida species compared to caspofungin and anidulafungin. The inhibitory effect of anidulafungin was higher for biofilms of Candida krusei, Candida tropicalis, and albicans compared to Candida paracelloses. In support of that, anidulafungin minimal inhibitory concentration for biofilms of Candida krusei, tropicalis, and albicans was 0.13 microgram per milliliter for each. The rest for the Candida paracelloses biofilms was 8 times higher. In summary, planktonic forms of Candida species were susceptible to anidulafungin, but susceptibility to caspofungin was observed in 85% of phthalate, to fluconazole in 67%. Minimal inhibitory concentration of conventional amphotericin B for Candida species was less than or equal to 1 microgram per milliliter. Both tested echinocandins were active in vitro against Candida biofilms. The maximum activity against Candida biofilms were obtained for anidulafungin, further for caspofungin. All tested Candida biofilms were resistant to fluconazole with minimal inhibitory concentration more than 1.024 microgram per milliliter, and conventional amphotericin B with MIC range from 4 to 16 microgram per milliliter. In conclusion, I want to thank all those who have helped in carrying out the research, among which Professor Klaseva Galina Alexandrovna and all my colleagues working in the Laboratory of Clinical Microbiology, Mycology and Antibiotic Treatment of National Research Center for Hematology in Moscow, Russian Federation. Thank you for your attention, and I'm sorry for some interruption with my slides. Thank you.